There are animals that can tear you apart, animals that can crush bones, animals that can kill with a single bite. But the most dangerous animal on Earth is something you could crush between your fingers. It weighs almost nothing, makes almost no sound, and yet, every year, it reaches more human beings than almost any predator ever could. It doesn't need strength. It doesn't need speed. It doesn't even need to fight you. It only needs a few seconds on your skin. Because the real weapon isn't the bite. It's what comes after it. And once you understand what is actually happening when a mosquito lands on you, you may never look at one the same way again. Because this isn't simply an insect. It is a biological delivery system. A grizzly bear relies on claws. A cobra carries venom. Yet, a fragile, weightless insect dwarfs their collective lethality. This creature does not overpower us. It does not physically attack. Instead, it acts as a vector, an organism that moves a pathogen from one host to another. But this threat remains highly conditional. Of the thousands of species, only a select few pose any danger, and then only if the individual insect is infected. Risk depends on the species, the pathogen, the region, and even the season. Most bites are harmless, a momentary irritation. But when the right variables align, what does that single bite actually set in motion? There is no single entity called the mosquito. Instead, there is a sprawling evolutionary tree of over 3,700 distinct species. Most are entirely harmless to us. The danger is highly specialized. 3 specific branches carry the weight of human mortality. First, the night-feeding Anopheles, the sole vector of malaria. Then, the urban-adapted Aedes, carrying dengue, yellow fever, and Zika. And Culex, quiet hosts to West Nile virus. Each branch has its own habitat, its own host preferences, and its own lethal passenger. But before any pathogen can be delivered, how does this fragile killer find us? The search begins with a breath. Every time we exhale, we release a plume of carbon dioxide. To a hungry female mosquito, this gas is not a waste product. It is an activation switch. From over 30 meters away, she detects the rising concentration of carbon dioxide. It gates her brain, triggering an immediate predatory search mode. She flies upwind, tracing the invisible current. At a range of a few meters, her sensory receptors refine the target. She tracks visual contrasts, dark shapes moving against the background. Then, she tunes into our skin chemistry. She smells a complex blend of volatile compounds, lactic acid, ammonia, non-anal, and short-chain carboxylic acids. Our sweat is a chemical signature, calling her in. But odor only gets her close. Within centimeters, the cues shift. She senses a sudden rise in humidity, the moisture evaporating from living skin. Then come the thermal gradients. She locks onto the infrared signature of our body heat, guiding her down to a precise landing zone. Different genera weigh these signals differently. While Aedes relies on the gating signal of carbon dioxide, Culex evaluates a broader, simultaneous mix of heat, shadow, and moisture. Yet, for all of them, the trap is set. The transmission has been received. She has landed. Both male and female mosquitoes survive on plant sugars and nectar, but the male's journey ends there. Hers demands a heavier toll. To develop her eggs, she requires a concentrated infusion of protein and lipids. Our blood triggers vitellogenesis, the biological process that synthesizes these nutrients into egg yolk. Over the next 3 to 5 days, she will digest this liquid meal, slowly forming up to 300 new lives. To her, we are simply a resource, a biological treasury. But to claim it, she must solve a critical problem: how to steal from us without being noticed. The proboscis is not a simple needle. It is a highly engineered, 6-part biological system. As she presses against the skin, the outer sheath retracts, releasing 6 microscopic stelates. 2 jagged maxillae act as saw blades, vibrating 15 times a second to slice the epidermis with almost zero resistance. 2 mandibles hold the tissue apart. Then, the needle-like labrum probes the subcutaneous darkness, its chemical receptors sniffing out a capillary. Once inside the vessel, a dual-channel flow begins. Through one channel, the hypopharynx pumps saliva down into the wound. This saliva is a sophisticated biochemical cocktail. Tachykinin peptides dilate local blood vessels. Apyrase prevents clotting. D7 proteins numb the pain receptors, keeping the host entirely unaware. Through the second channel, she draws blood upward. But this chemical exchange is a gateway. At this exact threshold, the fluid flowing into our bodies can carry far more than saliva. The blood meal is not a one-way transaction. If the host is infected, microscopic passengers slip into the mosquito's gut. Here, in a dark pool of digestive enzymes, a silent struggle begins. Most invaders would perish. But these specialized pathogens have evolved to survive. They penetrate the midgut wall, escaping into the insect's body cavity. Inside this fluid-filled space, the invader replicates, multiplying by the thousands. It is a slow, methodical colonization. Over several days, the pathogens migrate, steering directly toward the salivary glands. This delay, the extrinsic incubation period, is a biological clock. Its speed depends on the species, the pathogen, and the warmth of the surrounding air. Only when they invade the salivary duct is the transformation complete. The female mosquito is no longer just a feeder. She is a carrier. The next time she pierces skin to feed, the chemical exchange reverses. Along with her saliva, she pumps an active payload directly into the host's bloodstream. The mosquito is the delivery system. The pathogen is the real weapon. The night-feeding Anopheles mosquito hosts the oldest passenger of this biological design. When she feeds on an infected human, she draws up microscopic sexual stages of the parasite called gametocytes. Inside the cold, dark chamber of her gut, a sudden temperature drop and chemical cues trigger these cells to wake. Male gametocytes exflagellate, fertilizing the female gametes to produce a mobile, drilling organism known as an eukaryote. This microscopic traveler does not seek digestion. It seeks escape. The eukaryote bores directly through the mosquito's midgut wall, anchoring itself on the outer membrane to grow into a protective oocyst. Over the next 10 to 14 days, this cyst swells, maturing until it ruptures. Thousands of active, slender sporozoites spill into the insect's body fluid. Only a tiny fraction of these invaders survive the migration through her circulatory system to successfully invade her salivary glands, creating a tight biological bottleneck. Yet, the survivors that accumulate in the salivary duct are more than enough to infect the next host. The deadliest human malaria parasite to take this pathway is Plasmodium falciparum. Its global impact is mapped using mathematical models. Because tracking every single infection in remote regions is impossible, public health agencies must generate estimates with calculated uncertainty ranges, rather than relying on a perfect census. For 2024, the World Health Organization reported an estimated 282 million malaria cases and 610,000 deaths spanning 80 countries. This massive mortality burden is heavily concentrated. Approximately 95% of those estimated cases and deaths occur within the WHO African region. Within those borders, the vulnerability of youth becomes devastatingly clear. Children under 5 years old account for roughly 3/4 of the regional malaria deaths. A fragile insect carrying an invisible single-celled organism, turning a silent night bite into a global health crisis. Some vectors require the expansive wetlands of the wild. Aedes aegypti demands far less. It has engineered a domestic ecology, trading the remote jungle for the human neighborhood. To this species, our suburban backyards are not disruptions. They are ideal habitats. It does not seek natural lakes or open rivers. Instead, it breeds in the miniature, forgotten water sources of modern life: a discarded tire, a plastic bucket, a neglected flower pot, or a clogged roof gutter. A few milliliters of standing water is all it takes to complete a life cycle. It lives in our shadows, resting on our walls, waiting in our gardens. This predator does not need the wilderness. Sometimes, it only needs your neighborhood. In these urban shadows, Aedes aegypti circulates a different threat. Not a complex parasite, but a strip of genetic code. A virus. Dengue. In 2024, national surveillance systems captured the scale of this urban expansion, recording more than 14.4 million reported cases and 11,201 deaths. Unlike the modeled mathematical estimates used to track malaria, these figures reflect actual, documented surveillance. The virus is a shapeshifter, existing as 4 distinct serotypes: DEN1, DEN2, DEN3, and DEN4. An initial infection grants lifetime immunity to that specific strain, but leaves the host vulnerable to the other 3. In fact, a subsequent encounter with a different serotype can trigger a biological trap. Under certain conditions, old antibodies fail to neutralize the new invader. Instead, they bind to it, actively assisting the virus as it invades host cells. This process is called antibody-dependent enhancement. While this interaction elevates the risk of severe disease, a critical outcome is never guaranteed. For many, it is a painful fever. For some, the immune response itself becomes the danger. This biological network extends far beyond 2 diseases. Along the Aedes lineage, the same urban vectors carry yellow fever, Zika, and chikungunya. In separate, quieter habitats, the Culex genus circulates West Nile virus and Japanese encephalitis. There are even specialized pathways for parasitic worms causing lymphatic filariasis, and the abrupt outbreaks of Rift Valley fever. These pathogens are never interchangeable. Each requires a highly specific insect key. Yet together, they represent a vast, interconnected global threat. One family of fragile insects, multiple distinct pathogens, millions of infections across nearly every continent. What allows such a sprawling network to persist? Their persistence relies on rapid, constant replacement rather than individual strength. A single female can produce up to 300 eggs after just one blood meal. Instead of vast lakes, she seeks the most microscopic pockets of standing water, a discarded tire, a wet bottle cap, or a muddy footprint. These tiny reservoirs are free from the fish and predators that would easily consume her offspring. Within these protected nurseries, the transformation is swift. The egg becomes a larva, matures to a pupa, and emerges as an adult. Under favorable conditions, some Aedes populations complete this entire 4-stage cycle in just 7 to 10 days. Once they emerge, the clock ticks fast. An adult female's lifespan is short, generally lasting only 2 to 4 weeks. Yet this brief window is highly effective. It provides enough time for repeated blood feedings, pathogen incubation, and the sowing of the next generation, whether indoors or out. This evolutionary efficiency guarantees survival without requiring invincibility. They survive by outbreeding their own mortality. The blueprint of this survival is not fixed. It shifts with the air. Future disease transmission behaves as an interacting model of volatile factors rather than a fixed prediction. Temperature acts as a primary regulator. Moderate warmth accelerates larval growth and shortens the pathogen's incubation clock. But there is a physical ceiling. Extreme heat or cold reduces adult survival, halting the cycle entirely. Humidity further dictates these odds, either sustaining adult life or drying it out. Water and human movement complicate the equation. Heavy rains and infrastructure-dependent drainage failures produce temporary habitats, while dry spells trigger a different response. In droughts, people store water in open domestic containers, creating artificial nurseries. Global travel and trade carry eggs and larvae across borders, introducing vectors to new zones. Yet, human exposure remains heavily modified. Window screens, air conditioning, housing quality, and public health infrastructure stand between the insect and the host, and pathogens must be actively circulating to spark an outbreak. Each vector genus responds differently. Urban Aedes exploit concrete habitats. Malaria-linked anopheles track rural ecologies. Culex pursues separate temperate paths. To counter them, new biological controls are being deployed, though none are universal. In verified settings, Wolbachia-based programs are operational, arresting viral replication. Other tools, like sterile male releases, are strictly in the pilot stage, while genetic control research remains experimental. There is no simple victory. Insecticide resistance spreads, and changing environments constantly redefine the map. The system adapts, leaving the future highly undecided. For all its lethal success, the mosquito remains incredibly fragile. A gentle breeze, just 15 kilometers per hour, completely halts its flight. Extreme cold freezes its development, while dry heat quickly dehydrates its delicate body, causing rapid mortality. These physical boundaries are what humanity must exploit. Yet, there is no single, universal shield. A strategy designed for one vector is useless against another. The malaria-carrying Anopheles hunts indoors, under the cover of night. To stop it, we rely on insecticide-treated bed nets and residual wall sprays, intercepting the insect when it attempts to rest. But Aedes aegypti, the vector of dengue, behaves differently. It bites during the day, outdoors, and breeds in tiny domestic pockets of water. Bed nets cannot protect against a daytime bite. To stop Aedes, communities must eliminate the standing water in flower pots, gutters, and discarded tires. Prevention is not about making every mosquito disappear. It is about interrupting contact through precise, targeted action. This creature does not dominate through physical power. It has no claws, no crushing jaws. It is easily swept away by a sudden draft. Its true danger lies in its biology, a quiet, microscopic efficiency that turns a few brief seconds on human skin into global disease spread. We build walls, we rewrite genes, yet the system adapts. As our world warms, the boundaries of these silent pathogens expand. The ultimate threat to humanity is not a powerful predator, but a fragile, ancient network waiting just beyond the threshold of our homes. It only requires a single, quiet moment. What do you think? Share your thoughts below.